Hello, I'm Saurabh Sinha from the University of Illinois at Urbana, Champaign in the US, and it is my great pleasure to be talking to you today. Today, I'll present to you an overview of the NIH-funded Center for Excellence for Big Data to Knowledge, or BD2K, that I directed from 2014 to 2019, and which tackled the challenge of building powerful tools for genomics. I'll start by pointing you to this perspective piece that we wrote in 2015, where we recognized the amazing growth of data in genomics and predicted that soon genomics may become the biggest source of data for analysts. The funding agencies in the US are, of course, well aware of that and, in fact, pushing for this very vision and preparing the research community for it. For instance, there is considerable buy-in for the idea of fair data and software, where fair refers to findable, accessible, interoperable, and reusable. And among other ways to achieve the FAIR vision, there is a push for cloud-based software portals that can interoperate with each other. It is precisely this vision that drove the center that I'm going to talk to you about today. This BD2K Center was hosted at the Institute for Genomic Biology at UIUC in partnership with the Mayo Clinic at Rochester, Minnesota. I co-directed it with my colleague, Professor Jiawei Han, and I led the research team of the center. Our goal was to build a knowledge engine for genomics, from which the center got its name, Knowing. The main deliverable of the Knowing Center was the eponymous Knowing platform, a cloud-based portal for genomics data mining. The idea was that genomics data sets are often in the form of spreadsheets where rows represent genes and columns represent different conditions or samples under which the high throughput measurements have been made. The platform for analysis of such data is hosted on the Amazon cloud and allows the user to upload their own spreadsheet of genomics data, just as you would upload your photos to Google Photos, let's say. And then the user can run powerful analytic tools through the platform on the cloud. The tools offered by the Knowing system address a variety of popular bioinformatic tasks, such as sample clustering, gene set characterization, gene prioritization, phenotype prediction, signature analysis, and so on. And these tasks can be useful in answering common biological questions, such as what subtypes does this disease have? Which biological processes are these genes involved in? Which genes are associated with this phenotype? How long do we expect a patient to live? And so on. And the tools provided relied upon a variety of methods from machine learning and data mining. One of the highlights of Knowing analysis includes new tools for knowledge-guided analysis, whereby existing genomics knowledge bases or databases represented as a massive heterogeneous network are brought to bear upon the analytical operations provided to users. For this, we encode into a graph format the data from a number of genomics databases. These include pairwise relationships among genes, such as protein-protein interactions, genetic interactions, and homology, and so on, as well as annotations of genes, such as pathways, gene ontology, etc. The nodes of this graph will thus be the genes and properties, and the edges will represent the known gene-gene relationships and gene property annotations. And just to remind you, there's a lot of public data to encode, so the graph is quite large, with hundreds of thousands of nodes and 500 million edges. Now, here's the underlying idea of knowledge-guided analysis or knowledge network-guided analysis. You have this spreadsheet on the right with genes as rows and biological samples as columns, a typical input for machine learning algorithms. But you also have this separate graph from prior knowledge, where the nodes are the features of your spreadsheet. And the edges, therefore, represent relationships among these features. How can you exploit this graphical knowledge in improving the accuracy of your machine learning tools. Note that if there is one thing we've learned in 3 decades or more of bioinformatics, it is that prior knowledge and context really matter for machine learning performance. So this was the central algorithmic question driving our work: the use of knowledge network in the machine learning analysis of user spreadsheets. There's also an obvious cyber infrastructural challenge posed by knowledge-guided analysis. There's a massive knowledge network involved in the analysis, and we don't want users to have to download this to their computer and keep it updated all the time in order to run our machine learning tools on their data. Plus, some of the analysis tools may be prohibitively compute intensive, making local execution of the software not such an attractive prospect. The solution we pursued, therefore, was to have the knowledge network and the tools co-localize on the cloud, and the user simply moves their smaller data sets, the spreadsheets, to the cloud prior to analysis. We put together a strong software engineering team to make this happen, thanks to our partners at the National Center for Supercomputing Applications, or NCSA, and they made sure that the tools made by our graduate students and postdocs were suitably packaged in Docker containers and plugged into a Kubernetes orchestrated system for automated deployment and scaling. So this is what the data flow looks like for analysis at the Knowing platform. The researchers get their omics data from the biotech or sequencing center next to them, hopefully already formatted as a spreadsheet, and then they send this spreadsheet to the cloud where the knowledge network and the Knowing tools or pipelines already reside. The analysis is done at a click of a few buttons, and the results are sent back to the user. But there are other ways in which the Knowing tools can be run in the cloud. For instance, there is a cloud-based ecosystem of data and tools for cancer genomics, the CGC, or Cancer Genomics Cloud. That's this pink shaded cloud. The user may be doing their routine analysis on CGC datasets and say they want to run a Knowing tool, this one, then and there. For this, we have published our tools on the CGC, meaning that the tools are available for use on the CGC cloud, and they can seamlessly access the knowledge network, which resides in the Knowing cloud, thereby achieving knowledge-guided analysis without the user worrying about the logistics of moving the network or their data around. In the remaining time, I will briefly outline some of the bioinformatics approaches, the machine learning techniques that were developed by the Knowing Center. These pertained to the challenges of incorporating the knowledge network into their analysis, as well as to extract novel insights from heterogeneous multi-omics data. In one project, we were interested in identifying genes associated with drug resistance based on gene expression and cytotoxicity data on large panels of cell lines. Note that one can measure the sensitivity of a cell line to a drug, i.e. the cytotoxicity, by experimentally obtaining a dosage response curve such as this, and then summarizing it into a single number. Our collaborators at Mayo Clinic had such cytotoxicity data for many different drugs on cell lines derived from 300 different individuals of 3 different ethnicities. Moreover, gene expression data were also available on those cell lines. The simplest approach to identifying genes related to drug response variation among those individuals would be to use correlative methods between expression and cytotoxicity. But we showed the power of knowledge-guided analysis here. We transformed the gene expression matrix to incorporate the knowledge network using random walks with restarts or network diffusion, and then correlated the resulting transformed expression profiles of genes with the cytotoxicity profiles of cell lines to rank the genes. We showed that this clearly improves the ranking of genes compared to regularization-based approaches, which is what might come to your mind first, such as ElasticNet, without the use of knowledge networks. The top predictions of cytotoxicity-related genes were then experimentally validated by our collaborators. Another challenge where such a network diffusion approach made a big difference was that of sample clustering, where the goal is to cluster the mutation profiles of patients into groups in the hope of discovering subtypes of a disease. We adopted an approach published earlier by the Eidecker lab at UCSD for this purpose, where network diffusion is used to smooth the mutation profiles of patients before clustering them in using non-negative matrix factorization. The result is a powerful sample clustering tool where you upload a mutation matrix of genes by patients, and it will group the patients in a way that distinguishes different subtypes. So each of these blocks here of a different color is a different group of patients that was discovered in the cancer mutation profile data. In a different project, we tackled the challenge of defining gene signatures, which is a concept quite popular in pharmacogenomics and cancer. In particular, we asked if the transcriptomic signatures or transcriptomic changes due to a gene's knockdown in a cell line truly form a signature. We analyzed the Link's L1000 compendium of hundreds of thousands of such transcriptomic profiles and set up a classification task of predicting which gene's knockdown is represented by a given before and after view of the transcriptome. We used a convolutional neural network for this task and tested it on a 400-way classification task, where the expected accuracy is a mere 0.25%, and we achieved a 70% accuracy for this task in predicting which of 400 possible gene knockdowns the given transcriptomic change represents. In another project on cancer pharmacogenomics data, we used a probabilistic graphical model to integrate expression, genotype, i.e. EQTLs, epigenotype, i.e. EQDMs, and drug response data on a panel of cell lines, along with transcription factor ChIP-seq data from the ENCODE project, to identify major regulators or drivers of drug response variation across individuals. Again, our collaborators validated several of our predicted regulators via knockdowns in cell lines. In another work, we used a probabilistic graphical model to identify transcription factor gene regulatory relationships that especially matter to a given phenotypic variation, in this case, the tissue of origin of tumors, developed fast methods to identify pairs of genomic features for separating two classes of objects, such as up and down regulated genes, achieving over 400x speedups, developed novel ways to identify pathways associated with drug response using network diffusion again to represent genes and pathways in the same low-dimensional space, and made some progress on using models trained on cell line data to make drug response predictions on patient cohorts from TCGA. The culmination of those 5 years of work on building tools in cyber infrastructure is the Knowing platform, which allows users to run our tools in the cloud. In ending, I'd like to say that the genomics community has really got its act together in moving towards a fair world with easy-to-use tools and data, and our work was a small contribution to this community effort. Machine learning and AI will clearly play a key role in this analysis, given the high dimensionality of the data, the large volumes of training data, and the interrelationships among different components of the data. Large specialized databases are a trend, and these databases tend to be on the cloud these days. And the future tool development should be keenly aware of the need for interoperating with these cloud-based databases. Prior knowledge is key to achieving greater generalizability from machine learning techniques for biological data, and rigorous algorithmic approaches are necessary to incorporate that prior knowledge. And the grand challenge will therefore be to facilitate multi-level mechanism-driven analysis of multi-omics datasets informed by knowledge bases of molecular properties and interactions, and all of this on the cloud. I'd like to thank the NIH and an outstanding team of researchers, scientists, and software engineers at the University of Illinois and the Mayo Clinic for making this work possible. And I'd also like to thank Dr. Wasim Haider for inviting me to this fantastic symposium today. I hope you have a great day. Thanks.